Hello students, today I will give you a brief explanation about the cytoskeleton in the cell chapter. Let us see what are the components of cytoskeleton, what are their functions, what is the structure, all those details. In the cytoskeleton, the skeletal system which is present inside the cell is basically called a cytoskeleton. Let us see the components. The first components present in the cytoskeleton are microtubules. These are the largest components. With 25 nanometers diameter. Among all the cell components which are present in the cytoskeleton, the largest ones are the microtubules. They have 25 nanometers diameter, and these are basically made up of tubulin protein. They are made up of proteins only, and that particular protein which is forming the microtubule is the tubulin protein. Along with that, they will be having in cilia and flagella, they will be having dynein protein. But the main protein which is present in the microtubules is the tubulin protein. And these microtubules, they will be forming different structures like cilia, flagella, centrosome, basal body, spindle at the time of cell division. These are all formed by the microtubules only. In this way, the microtubules will be helping the cell in order of motility. If the cell is able to move from one place to another place, that locomotory structures are cilia and flagella in some of the cells, that movement is provided by the microtubules through cilia and flagella. Then centrosome, which helps in cell division, that is also made up of microtubules. Then basal body, which is the basal part of the flagellum or the cilium, from where the flagellum arises, that part is called as a basal body, that is also made up of microtubules. Spindle form, spindle which is helping in the chromosomal movement at the time of cell division, that is also made up of microtubules. So these are all the basic structures which are formed by the microtubules. In this way, the microtubules basically help in the motility, and sometimes they help even in the maintenance of the shape of the cell, and they also help in the cell division in the formation of the spindle. And these microtubules are generally located in the cytoplasm only. The next one, intermediate filaments. These are the second components, and these are having the intermediate size, medium size in between the microtubules and the third component, microfilaments. These are having a diameter of 8 to 12 nanometers. 8 to 12 nanometers is the diameter, and these are made up of proteins. Different proteins are there: vimentin, keratin, desmin, lamin, proteins, etc. These are all different kinds of proteins which are present in the intermediate filaments. So most heterogeneous type of cytoskeletal elements are the intermediate filaments, and these are like almost more stable when compared to the microtubules. Microtubules are hollow cylinders, whereas these are somewhat like stable rod shaped structures, and they are formed by the different protein fibers which are coiled or twisted among themselves to form the rope like structures, rope like fibers, because they are twisted. They are more stable, and they are basically non-polarized ones. And these intermediate filaments, they are generally present in the cytoplasm as well as in the nucleus. Present in nucleus as well as in the cytoplasm. In terms of nucleus, they help in the formation of nuclear lamina. They are meant for anchorage of the nucleus, and then anchorage of the chromosomes during the cell division. And in the cytoplasm, they are meant for cell to cell connections like desmosomes. They will be connecting and they will be anchoring to the extracellular matrix, anchorage of the nucleus, cell organelles, extracellular matrix, that is provided by the intermediate filaments. They help in the maintenance of the shape of the cell, and they help in positioning of the nucleus and various cell organelles in their place, and they will be connecting one cell to the other cell in terms of the extracellular matrix connections, desmosomal connections, etc. These are the basic functions of intermediate filaments. Next, the last component of the cytoskeleton are microfilaments. These are the smallest among all the three cytoskeletal elements, and they will be having a diameter of 7 nanometers diameter, with an average, basically 6 to 8 nanometers will be the diameter, but with an average of 7 nanometers. And these microfilaments, they are made up of actin protein. Globular actin protein will be there, and they will be forming the filamentous actin protein. So, mainly actin protein will be there, and actin protein globular, it will become the filamentous one. So, the filaments are made up of actin protein. These are generally present in the muscle cells, in all the animal cells these are present. Okay. Basically, all the cytoskeletal elements are present in all the eukaryotes, but in case of intermediate filaments, they are present only in the animal cells, whereas microtubules and microfilaments, they are present in the plant and animal cells. These are also solid structures only, because they are having the globular actin, so solid structures. And these microfilaments, they will be helping in the muscles they are present, so they are also called as myofilaments. They associate with myosin, and they help in muscle contraction. And pseudopodia formation. They help in the pseudopodia formation because they lie below the plasma membrane. So, they are basically located in the cytoplasm only, in cytoplasm. Pseudopodia will be formed, and they help in maintenance of the cell shape and even the cell motility. Maintenance of the cell shape and cell motility in terms of cytoplasmic streaming. Cytoplasm of a cell will be moving with the help of microfilaments, which is also called as cyclosis. So, microfilaments, they help in muscle contraction, cytoplasmic streaming, pseudopodia formation, maintenance of the cell shape. These are present only in the cytoplasm, not present in the nucleus. So, basically cytoskeleton is made up of three components: microtubules, intermediate filaments and microfilaments. Microtubules, microfilaments, they are present in the cytoplasm. Intermediate filaments are present in both nucleus and cytoplasm. Only intermediate filaments are present in the animal cells, that too some of the animal cells only. But microtubules and microfilaments are present in both plant and animal cells. In all eukaryotes, they are present. But cytoskeleton, before we used to think that it is the characteristic features of only eukaryotes. But right now we came to know that even the prokaryotes like bacteria also possess similar structures like that of the microtubules, microfilaments, etc. So, now cytoskeleton is present even in the prokaryotes like bacteria. It is not found, we used to think, but that is not correct. So, cytoskeleton is present even in the prokaryotes. With this, the explanation part of the cytoskeleton is over, which is made up of three components. Let us see the next one, the questions. The first question: cytoskeleton in a cell is involved in functions like, which functions are performed by the cytoskeleton? The first one, mechanical support, motility and maintenance of the shape of the cell. These three are the main functions which are given in the NCERT book. So, all these three are correct. So, the correct option for this question will be Cytoskeleton in a cell is involved in the functions like mechanical support, motility and maintenance of the shape of this cell. Let us see the next one. Cytoskeleton refers to the cilia, flagella only, microtubules only, network of filamentous proteinaceous structures, both B and C. So, in this the only is incorrect. Anyhow, cytoskeleton refers to not cilia and flagella, it refers to microtubules, but microtubules will be forming the cilia and flagella. So, this is not correct. Microtubules only, this is also not correct, but network of filamentous proteinaceous structures, it is correct, because all that microtubules, intermediate filaments and microfilaments, they will be arranged in the form of a reticulate network like structures throughout the cell. So, they will be giving a particular maintenance of the shape of the cell is done by these three: microtubules, microfilaments and intermediate filaments. So, cytoskeleton refers to network of filamentous proteinaceous structures is correct, not both B and C. Microtubules and microfilaments, intermediate filaments, are constituents of central sheath. This is present in the centrosome, in the central part. Centrosome is made up of two centrioles. Okay, in the centrioles, central sheath is present. Cytolamellae, lamellar structures of this cell. Then cytoskeleton. You know that the cytoskeleton is made up of three components: microtubules, microfilaments and intermediate filaments. So, for this the correct option would be cytoskeleton, because it is composed of the three constituents like those three. Which of the following are found only in animal cells? Only in animal cells. This is the clue for this question. Microtubules are present in both plant and animal cells. Microfilaments are also present in plant and animal cells, but whereas intermediate filaments, they are present only in animal cells. So, for this question the correct option will be the intermediate filaments. Microfilaments are composed of tubulin, myrosin, niacin and actin. Tubulin is present in the microtubules. Myrosin is an enzyme. Niacin is a vitamin. Actin, it is present in the microfilaments. So, for this question the correct option will be actin. Next one. Cytoskeletal filaments are polymers of, they are made up of which monomeric units? Are they made up of carbohydrates, ribonucleotides, proteins, deoxyribonucleotides? In this, the cytoskeletal elements are basically made up of proteins. They are never made up of carbohydrates, and this ribonucleotides polymers will be RNA, deoxyribonucleotides polymers will be DNA. So, cytoskeletal filaments are all made up of only proteins. Next one. Cytoskeleton is absent in eukaryotic plant cells, prokaryotic bacterial cells, eukaryotic animal cells, none of the above. You know that recently they found that even in prokaryotes also cytoskeleton is present, or similar structures like that of the cytoskeleton are present. So, for this question, in eukaryotes, in all eukaryotes anyhow they are present, because cytoskeleton is a unique feature used to think. So, now they found out even in prokaryotes, in this also it is present, in this also it is present, in this also it is present. So, for this question the answer will be none of these. Which is incorrect with respect to functions of microfilaments? Microfilaments, which is incorrect? This you have to find out. The cyclosis is also called as cytoplasmic streaming. Then furrow formation, it helps in cell division or cytokinesis process. Then pseudopodia formation, that also is a function of microfilament. Spindle formation, this is the function of microtubules. So, for this question, spindle is formed only by the microtubules, not by the microfilaments. So, for this question the correct answer will be spindle formation. Next one, most heterogeneous type of cytoskeletal filaments are. Heterogeneous means which are made up of all different kinds of substances, basically proteins. So microtubules are made up of only tubulin protein, tubulin main protein. Microfilaments are made up of actin. Intermediate filaments are made up of keratin, vimentin, desmin and lamin, etc. So here which is made up of different types of proteins, basically intermediate filaments. So for this question the correct answer will be intermediate filaments. Find the incorrect statement. Microtubules, they form the cilia. This is correct only. In the explanation part I have told you already. Microfilaments are not made up of tubulin. This is also correct because microfilaments are made up of actin. So they are not made up of tubulin. That is also correct. Intermediate filaments are resistant to colchicine. They are resistant to colchicine, intermediate filaments, but microtubules are resistant to colchicine. This is wrong because microtubules are sensitive to colchicine. Microtubules will be arrested by the colchicine because it is obtained from a particular plant and which will arrest the cell division in the metaphase without forming the spindle. So if you want to stop the cell division by arresting the cell division before the formation of spindle or spindle formation should be affected, means we have to use the colchicine because colchicine will be affecting the microtubules by not forming the spindle. So in that way we can arrest the cell division because microtubules are sensitive to colchicine. So this statement is the incorrect statement. So for this question the correct option will be microtubules are resistant to colchicine is incorrect. Next one, microtubules are not found in, flagella they are found, in spindle also they are present, in cilia also they are found. In the nucleus they are not present. So they are not found inside the nucleus. As already I told you, in the nucleus only intermediate filaments are present. How many protofilaments are present in a mammalian microtubule? You know that in the centrosome 2 centrioles are present and each centriole is made up of 9 triplets of microtubules with the 9+0 condition in centrioles. In each centriole 9+0 arrangement of microtubules. In cilia and flagella, one cilia or one flagella of eukaryotes, we are talking about eukaryotes only, 9+2 arrangement of microtubules will be there, where that 9 microtubules will be doublets. So 18 plus 2, 20 microtubules will be there altogether here in cilia and flagella of eukaryotes, whereas in centriole, in one centriole of centrosome 27 will be there microtubules with 0 arrangement, 9+0 arrangement. In the center no microtubules are present here, but each microtubule, whatever this 9 we have written, that each microtubule it is made up of 3 subtubules, and in that each microtubule filament is made up of 13 protofilaments. So for this one the question answer is 13. 20, 27, 18 is wrong because each mammalian microtubule it is formed by 13 protofilaments. So they will be arranged together to form a hollow cylinder. 13 will be arranged together and it will be hollow. Whereas microfilaments and intermediate filaments generally they are solid. Neurofibrillary tangles found in the brain cells of persons with neurodegenerative disorders consists of. These neurodegenerative disorders are generally associated with microtubules, microtubule associated proteins, microfilaments, intermediate filaments. These are associated with microtubule proteins. These are having some defect in that neurodegenerative disorders showing patients. So, this neurological disorders like Alzheimer's disease, okay, that and all are related to the microtubule associated proteins. The microtubule binding activity of microtubule associated proteins is controlled by which process? Is it oxidation, reduction, hydrolysis and phosphorylation? You know that for the functionality, for the movement of a cell movement or cilia movement or flagella movement, we need energy in the form of ATP. Here also that microtubule binding activity of microtubule associated proteins, how they will be able to move because of phosphorylation. Phosphorylation is nothing but addition of phosphate. Addition of phosphate. So, by the phosphorylation process, they will be getting the energy for them to do particular activity. So, the main process which is involved here is phosphorylation. Malformation of these are linked to the inability of muscle contraction. You know that muscle contraction is one of the functions of the microfilaments. Centrioles, no. Microtubules, no. Intermediate filaments, no. Microfilaments, yes, because they are the ones which are present in the muscles, and that is why they are called as myofilaments, and they help in the muscle contraction. If these are malformed, then generally the contraction of the muscles will be affected. So, for this the answer will be microfilaments. Which cellular structures are responsible for the lack of motility? Lack of motility. Basically, the cell motility in terms of cilia and flagella are provided by the microtubules. But in terms of cell to cell movement or cytoplasmic streaming, pseudopodia formation, that is provided by the microfilaments. But never that movement will be provided by the intermediate filaments. So, ER is not associated with that, but lack of motility of this is not concerned with that. Microtubules, microfilaments, intermediate filaments. So, here if you consider the cilia and flagella, the answer will be microtubules. If microtubules are not there, in terms of cilia, flagella, the motility of a cell will not be happening, because these microtubules are needed for the cilia formation and flagella formation. If you consider only cytoplasmic streaming, or particularly if they give pseudopodia or cyclosis, then the microfilaments lack of this can be the reason for the lack of motility for the cell. So, for this I have taken the generalized motile organs like cilia and flagella, because those two will be formed by the microtubules. So, for this question the answer will be microtubules. Next one, microfilaments are involved in phagocytosis, pinocytosis, exocytosis, ephagy. Phagocytosis is a process of endocytosis, engulfing the food materials into the cell. That is done by the microfilaments, because they are present beneath the plasma membrane and they will be taking in the food materials or pathogens. Then pinocytosis is cell drinking, that is not done by the microfilaments. Exocytosis, throwing out the waste. Ephagy, throwing out the metabolic waste from the cell, that is also not done by the microfilaments. They help in phagocytosis. So, for this question the answer will be phagocytosis. In which of the following processes is the cytoskeleton involved? Contractile ring formed during cytogenesis. Contractile ring which is formed during the cell division, cell formation, new cell formation, that is actually done by the microfilaments. Anchoring the extracellular matrix, anchorage of nucleus, anchorage of cell organelles, this is done by the intermediate filaments. Then post-transcriptional modification of mRNA. This is done by certain proteins, but this is not a function of cytoskeletal elements. Post-translational modification of proteins, this is also done by certain enzymes and proteins, not by the cytoskeletal elements. So for this question, these two are correct, these two are wrong. So in the given options, A and B is there for the two. So second option would be correct for this. How many of the following structures are synthesized by microtubules? Centrioles, they are synthesized by the microtubules only. Cilia, they are synthesized by microtubules. Flagella, spindle and basal body. All these 5 are synthesized by the cytoskeletal elements only. So definitely 1, 2, 3, 4 and 5. So all the 5 are formed by the microtubules basically. So for this question, the correct answer will be 5. If cytoskeleton is missing, then the cell would lose its shape, structure, not the functions. So for this question, the answer will be the shape and structure will be altered if cytoskeleton is not there. In the NCERT, the functions like cell motility, maintenance of the shape of the cell and mechanical support are the 3 functions which are given in the functions. So shape and structure will be altered if the cytoskeleton is missing. With this, we have completed the cytoskeleton, and all the best for your tests.